Step into any gym and you'll hear the same guarded rule. Spot reduction is a myth, and doing endless crunches will not burn belly fat. But recent physiological studies indicate this unbreakable rule is actually biologically incomplete. To understand why, we have to look past the skin and examine the true layered architecture of the core itself. The midsection is a complex, multi-layered mechanical engine capable of directly manipulating its local environment. This diagram shows the first, most superficial muscle layer, the rectus abdominis. It flexes the torso and is commonly known as the six-pack. Deeper down are the second and third functional layers, the external and internal obliques, which control lateral rotation and bending. Beneath all of that lies the deepest layer, the transversus abdominis, acting as an internal corset for core stability. These deep muscles are dense and packed with mitochondria. They demand massive amounts of energy whenever they contract, whether curling upward or holding a static plank. This intense, localized energy demand creates a unique metabolic interaction with the stubborn adipose tissue resting directly on top of them. When your body needs energy, it initiates a process called lipolysis, breaking down stored triglycerides into usable free fatty acids. This process is triggered by stress hormones like epinephrine, which circulate through the bloodstream during exercise. Because these energy-demanding core stabilizers sit immediately beneath stubborn subcutaneous belly fat, they possess a proximity advantage. Intense, targeted core contraction increases localized blood flow, actively pulling more of those circulating, fat-mobilizing hormones directly into the abdominal area. The working core acts as a localized metabolic sink, preferentially breaking down the adjacent fat cells, simply because they are closest to the active muscle tissue. Researchers at the Copenhagen Muscle Research Center put this proximity advantage to the test in human subjects. This graph tracks interstitial glycerol, which is a direct marker of fat breakdown. Over time, there is a clear, widening gap. Glycerol levels were significantly higher in the subcutaneous fat adjacent to a contracting muscle compared to a resting muscle. Looking at this subsequent chart, the data shows that the total release of glycerol was definitively higher in the targeted areas across varying intensities, marked here by statistical significance at 85% max workload. The clinical data proves that spot lipolysis, the localized, targeted breakdown of fat, is a biological reality. Targeted core work successfully breaks the fat right out of the cells in your belly, challenging our traditional understanding of abdominal exercise dynamics. There is, however, a crucial physiological catch. Successfully breaking fat out of a belly cell is not exactly the same thing as burning it away. The underlying muscle tissue and subcutaneous fat are separated by a rigid anatomical barrier, each with separate blood and drainage systems. The freed fatty acids do not go straight into abdominal muscles for fuel. Instead, they drain directly into general bodily circulation. If your body's overall energy demand isn't high enough during the workout, those free-floating fatty acids will simply be restored as fat somewhere else in the body. High-intensity exercise training creates the massive, whole-body energy deficit that is required to permanently incinerate those circulating fatty acids. This leads to an optimal two-step workout strategy for a leaner midsection. Step 1. Use targeted isometric and concentric core exercises, like planks and crunches, to trigger spot lipolysis and free the stubborn belly fat from its cells. Step 2. Follow up immediately with high-intensity, full-body metabolic conditioning to actually burn those newly freed fatty acids for good. Pure spot reduction remains a half-truth, but strategically combining the mechanics of targeted core lipolysis with systemic metabolic demand provides an evidence-based method for transforming your midsection.